So as we talk about mitosis, we need to understand that in actuality, there's a cell cycle, and mitosis actually only makes up a small portion of that cell cycle. So you see here the entire cell cycle, and all of this makes up what we call interphase.
Now, what's left of the cell cycle is that small piece that's called the mitotic phase. And this includes mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm.
Mitosis is cell division, and we can think of lots of reasons that a cell might need to divide. So as an organism grows, as if damage occurs and we need repair, as cells become older, we need to replace those. So there's several reasons for cell division. And let's start by going through the process of interphase. So interphase is broken up into three separate components, G1, S, and G2. In G1, that's sort of the first growth or the first gap phase. And it doesn't mean that nothing is happening then. Actually, there are lots of things happening. For example, the individual building blocks that are necessary in the next phase are being accumulated and built up, as well as the cell is producing ATP energy that's going to be needed later on in the process. So after the first gap phase, we have S phase, which stands for DNA synthesis. So in this important phase, the entire genome, or all of the DNA, is copied. So that now there are 2 copies of everything in this cell. And that's in preparation because as this cell begins to divide, it needs to have 2 copies of every single strand of DNA or every single chromosome so that each cell is going to get sufficiently 1 full copy of chromosomes.
So we've looked at the cell cycle, and we know that during interphase, specifically S phase of interphase, is when the DNA gets completely copied or replicated so that the cell is ready to undergo cell division or mitosis. So just as a quick review, let's look at some DNA structure information, and then we'll talk about how the DNA is copied in S phase. So remember, DNA can exist in the cell as chromatin or chromosomes. So we see here the chromosomes are the most condensed form. Chromatin is less condensed. And remember that during interphase, DNA exists as chromatin, and only during cell division or mitosis does it exist as a chromosome. Now, I also wanted to just review that if we unwind the DNA from all the histones and we look at what we call naked DNA, that the DNA is basically double-stranded. One strand is running one direction, one is running the other, so they are in opposite directions. But the 2 strands are held together by hydrogen bonding between the nucleotides or the the nitrogenous bases. Adenine pairs with thymine, guanine pairs with cytosine, and they always pair in that specific way. Now, during DNA replication, what has to happen is that the double-stranded DNA that exists in the cell has to be unzipped or unwound so that the 2 existing strands are separated. This enzyme right here called helicase is going to do just that. It's going to unzip or unwind those 2 strands, and both of them are going to serve as a template. So each one is going to be copied. Now, the enzyme that is doing the copying is called DNA polymerase. Now, remember, ASE tells us it's an enzyme. Polymerase means to create a polymer, and so that means we're creating a DNA polymer. That's the job of the enzyme. Now, it's important that we see that both of the template strands are being copied by this DNA polymerase. Now, one strand is called the leading strand and one strand is called the lagging strand. And that's because, as I mentioned, these are running in different directions. And the DNA polymerase enzyme can only read DNA and make a copy in one direction. So one strand, the enzyme can do it in a continuous copy. The other strand, it has to make a short copy, jump off, jump back on. But the cell has a way to ligate those all together at the end. So ultimately, what the DNA polymerase enzyme will do is, as it reads the template, if it reads an adenine, it will add a thymine to the new strand. If it reads a cytosine, it will add a guanine. It will do that one nucleotide at a time till it has synthesized or made a new copy of the complete template strand. So at the end of this process, what we'll have is 2 complete sets of DNA. One set will be based on one template strand, so we'll have one original strand with one new strand. The other set will have the other template strand with one new strand. So we call that semi-conservative because every time DNA is replicated, one strand of DNA is the old template strand with one newly synthesized DNA strand.
Now, as we move on to the next phase, G2 or gap 2 or growth phase 2, there are going to be some things that the cell has to have in preparation for entering into mitosis. So for example, there's going to need to be a good amount of cytoskeleton around, as we'll see that's necessary in mitosis. There's also going to have to be not just a duplication of all the DNA, which occurred in S phase, but also of the cytoplasm and all of the organelles in the cytoplasm before the cell actually enters the mitotic phase. Now, we're going to spend some time in a minute talking about what actually goes on during mitosis, where one cell divides into 2. But first, I want us to spend a little time, before I move on though, I want to make sure I point something out to you. This portion, mitosis, is cell division. The DNA gets duplicated well before the cell enters into mitosis. And in fact, there's an error check there to make sure that everything has been copied before the cell can enter into cell division. Now we're going to be talking a lot about chromosomes as we go through the cell division process. I want to make sure we understand some of the anatomy of a chromosome and some of the terminology. So first of all, what we see here is actually a duplicated chromosome. So this chromosome has already gone through S phase because everything has been copied. In other words, a single chromosome, okay, would just have one arm like this. This would be an unduplicated chromosome. So we can actually divide this chromosome right down the middle and say, well, these are 2 sisters, okay, and they are exactly copies of one another. So this sister and this sister are identical because during the S phase process, the entire strand or all of the DNA nucleotides were copied, and that's why we have 2 sister chromatids. Now this middle region where it looks like sort of the belt, okay, of the chromosome is called the centromere. This is where the spindle fibers or the cytoskeleton that actually manipulates and moves the chromosomes around, this is where the spindles will actually attach to the chromosome. Now something else that I want us to talk about is something called homologous pairs. So this is one chromosome shown here. Okay, so imagine if these were exactly the same size, okay? And actually what I'm going to do is I'm going to make this chromosome be red, okay? And this one blue. Let's say this is a pair of chromosome 1. Okay, so these are the same chromosome. They have the same genes on them. They may not be the same form of the gene, but they have the same genes. This would be, okay, let's say the maternal chromosome, and this other one would be the paternal chromosome. The pair of these together is called a homologous pair. Okay, because they are the same chromosome. Now, if you had a chromosome 1 and a chromosome 5, they wouldn't be homologous pairs because they don't have the same genes on those chromosomes. Homologous pair can either be the duplicated chromosomes like we see here, or we can have an unduplicated pair of chromosomes, either way. Now we're going to move on and talk a little bit about mitosis. Now mitosis is divided up into phases, okay? Prophase, prometaphase, metaphase, anaphase, and telophase or telophase. So I remember this with PMAT, P-M-A-T, and just remember that prometaphase is in there in between prophase and metaphase. Now remember, this is that small piece of the pie in the cell cycle where the actual cell division begins. And what's the goal of mitosis? It's because this cell that's about to divide has duplicated, right? It's got double the DNA, it's got double the cytoplasm, it's got double the organelles. So we're just separating, or the cell is just separating into two identical daughter cells. So genetically they will be identical because we just had a complete copy made of all the chromosomes in the cell. Now there's some things that we should know about the phases. So for example, in prophase, what do we expect to happen? Well, the DNA, the form of DNA, which when a cell is not in cell division, exists as chromatin. So it's not quite as tightly packaged as it would be in a chromosome. When the cell enters into cell division, then the DNA becomes packaged in its tightest form as a chromosome. So we begin to see visible chromosomes as the DNA condenses. We also begin to see the spindle fiber. So this is this apparatus made up of microtubules, so part of the cytoskeleton in the cell. And these are going to be responsible for attaching to and moving, manipulating the chromosomes so that everything arrives where it should, right, to the correct daughter cell. We're going to see the nuclear envelope begin to disappear, go away, as well as the nucleolus. So as we move on to prometaphase, this process just continues. Remember, the cell, this is a continuous process for the cell. We're just marking as certain things happen, we're saying we call this portion of mitosis. So in prometaphase, we see that the spindle is attaching to those kinetochore proteins of the centromere. So that's sort of that waist-looking area where the belt might be in the chromosome. Okay, the chromosomes will continue to condense even further. For metaphase, what we recognize, and you can see from this image here, the chromosomes line up in the middle or the equator of the cell. So they line up single file, and you see that the spindle fibers, right, are attaching to the centromeres of the chromosomes. Now at this point, the sister chromatids are still attached, right? We have the duplicated chromosomes intact, lined up in the equator of the cell. In anaphase, what you'll notice is the sister chromatids have separated, okay, they're no longer attached, and now they are sister chromosomes. So when they're attached together, each arm is called a sister chromatid. Once they're separate, now they are sister chromosomes. Okay, now the spindle fibers are actually moving those sister chromosomes to opposite poles or opposite ends of the cell. In telophase, we see that the chromosomes actually arrive at the opposite ends, and essentially the opposite of prophase and prometaphase begin to occur. So we see that the nuclear envelope begins to reform, the spindle begins to break down, and ultimately, what we've been tracking is the movement of the chromosomes, right? What's going on in the nucleus. But ultimately, for cell division to be complete, we have to end with cytokinesis, or the division of the cytoplasm into the two distinct cells. And it happens differently for plant or animal cells. So the one pictured here is like an animal cell where it just begins to pinch in, okay, which is called a cleavage furrow, because animal cells don't have a cell wall, a rigid cell wall.